Muy bien, muy buenos días. Siendo las 9 de la mañana, una vez más les damos la bienvenida a este curso acerca del valor de inmunomodular en cáncer. La presentación que tenemos para hoy es una presentación especial. Estamos dando inicio al tercer módulo de este curso y lo vamos a hacer por todo lo alto. Voy a hablar ahora mismo en español, después voy a hablar en inglés para hacer la presentación de la doctora Sanaz Taromi, que estará con nosotros. Sanaz viene, al igual que el doctor Carlos Panero, viene directamente de la empresa en Alemania y nos viene a tocar aspectos importantes acerca de las evidencias preclínicas y clínicas de Helixor en el contexto del cáncer, particularmente su relación con los componentes del sistema inmune. Voy a presentar a la doctora Sanaz. Voy a leer algunos aspectos básicos que ella me compartió para la presentación. La doctora Sanaz Taromi es biólogo molecular y es, digamos, asesora médica senior de la empresa Helixor. Antes de trabajar en Helixor, ella estuvo 18 años en el centro médico de la Universidad de Freiburg, donde lideró un grupo de investigación y además actuaba como profesora en la universidad. Su aspecto fundamental, el foco fundamental de su investigación, en la parte de investigación más básica, digamos, estuvo relacionada con el desarrollo de terapias CAR-T-cells, que es una estrategia de inmunoterapia para cáncer que seguramente han escuchado y de hecho la tratamos acá en el curso en algún momento, particularmente para el tratamiento de tumores sólidos, que es muy peculiar porque esta terapia sobre todo está enfocada en neoplasias hematológicas y, bueno, en general ha estado vinculada con el desarrollo de estrategias de terapia celular en el contexto del cáncer. Después de esta presentación, now I will speak in English to properly introduce Dr. Sanaz Taromi. Thanks a lot for your time. Thanks a lot for giving us the opportunity to have you in this meeting. And this is extremely important for us to receive direct information from Helixor regarding what is Helixor doing on cancer patients, mainly its interaction with the immune system. And this is one of the main topics that we are attending on this meeting. So for us, it's very important to having you here. Thank you so much for your time. And I'm pretty sure that this will be an excellent presentation and we will have an excellent information regarding Helixor. So I will activate the translation so we will hear Sanaz in Spanish. Wait a second, please, to activate it. And then once I do it, you can start. 1 second, please. Español. So, should be ready. Sanaz, microphones belongs to you.
Wonderful. Thank you very much. Thanks a lot, dear Joel, for the very nice introduction.
Sorry. Remember that you have to activate to say that you agree with the translation.
The translation is
It's perfect. It's perfect.
It's working now? Wonderful. Okay. So if anything goes wrong, just let me know. Thank you very much, dear Joel, for the nice introduction. And good afternoon or good evening, dear colleagues. I really feel honored to talk to you about immunological constants and mechanisms of action related to Helixor viscum album. In my current research, I focus on the immunological concepts and mechanisms of action of viscum album. Why? Because I have always been fascinated by the concept of holism and the idea of viewing biological processes as part of an interconnection whole. So today I am going to present you Helixor as an immunomodulator in cancer. What do we know and what remains to be explored? Main topics are general mechanisms of action, especially regarding Helixor viscum album. I will introduce it with different hallmarks of cancer Then we will go together into the immunomodulatory effects. Here, I would like to emphasize NK cells because they are so important in cancer research. Very shortly, HPV positive patients, just to show you that it has also antiviral effects. And then in the second half, I will introduce you clinical studies showing the safety and tolerability of Discom album in combination with immune checkpoint inhibitors, and afterwards the clinical efficacy, specifically overall survival. I will then, at the end of the presentation, share with you the case report. It is not published yet, but these are very interesting preliminary data from a very fantastic colleague who treated metastasis with Discom album monotherapy. So let's get started. For me, having been shaped by 5 different cultures, I have learned that progress does not arise from separation, but from connection and integrating different ways of thinking. And I had always thought this because in modern science, we break down complex phenomena into their individual components, and by doing so, we have achieved, of course, remarkable progress, right? We identified signaling pathways, we characterized receptors, we sequenced genes, and this is also what I did 18 years in my research as on CAR T cells, as Dr. Joel mentioned. However, as you see here, cancer does not arise from a single altered signaling pathway, right? It is a systemic disease. It develops through disruption of multiple interconnected levels, which means that tumor cells communicate with each other, but not only with each other, they also communicate with stromal cells, they remodel extracellular matrix, they manipulate blood vessels, they alter metabolic processes, and they evade the immune system. And I think perhaps this also explains why the concept of hallmarks of cancer has had such a profound impact on cancer research. You may know the paper of Hanahan and Weinberg who did not define cancer as a single mutation or a single mechanism. Rather, they described it as an acquisition of multiple biological capabilities that together give rise to the malignant state, which means that cancer emerges from a network, from interactions, from the system as a whole. And these hallmarks include sustaining proliferative signaling, we have a resisting programmed cell death, we have inducing or assessing vasculature, which means genesis, unlocking plasmatic plasticity, activating invasion and metastasis, which in my opinion is a very important, the most important point, actually, step in cancer therapy. Evading immune destruction, here I would like to go much further because this album plays here a huge role. Deregulating cellular metabolism, establishing replicative immortality, and inactivating growth suppressors. As I mentioned, remarkably, a growing body of preclinical but also clinical evidence suggests that this album extracts may directly or indirectly modulate each of these biological hallmarks, and this is precisely why Mr. Therapy fascinates me from a scientific perspective of view. With most interventions, we encounter there is a single logic, which I did always. One target molecule, one signaling pathway, one effect. With Discom album, however, a different picture emerges. We appear to be dealing with a therapy that acts simultaneously at multiple levels. It can inhibit cell proliferation and diffuse apoptosis. It can suppress angiogenesis, influence cell phenotype, not only tumor cells, but also tumor microenvironment cell phenotypes, and thereby even modulate tumor invasion and metastasis. And it may help normalize and alter metabolism of tumor cells. Let's say Warburg effect, right? So it can activate both the innate and adaptive immune system, and so on and so on. This means that these hormone interactions inhibit both tumor inhibitory and immune stimulating effect. So they show two huge systems simultaneously in both directions. Inhibition of tumor cells, cytotoxicity, but on the same side, they activate the immune system. Let's look at it a little bit further. So their inhibitory activity is mediated through the induction of apoptosis, and consequently...
Sorry, Sanaz, sorry.
No,
there is a problem with your microphones. Your microphone probably is better if you use your headphone. Because
you don't hear me well?
It's like it's not completely clear,
the translation.
No, not that, I mean the translation, but because of the audio.
Let's do it this way. I'm sorry. Do you hear me?
Yes, better now. Now, speak, please. No, but you, yes. Speak now, please.
Is it better now? I hope,
Perfect.
because I hear you very well.
Yes,
Wonderful.
Perfect. It's much better. I will
Okay.
activate the translation again. Sorry,
Yes.
sorry.
No worries. I'm sorry. Thank you for letting me know.
Yeah. Perfect. Excellent.
Please interrupt anytime it's needed. Thank you very much. Okay, so we stopped at the effect on tumor cells. We said that Viscum album has effect on tumor cells, like cytotoxic effect, but also on immune cells. One is inhibitory, the other one is activatory at the same time simultaneously. So, as I mentioned, the tumor inhibitory effect is first cytotoxic effect that ultimately leads to growth inhibition of the tumor and tumor reduction. This can occur directly through the cytotoxic activity, including effects on therapy-resistant cancer cells. This is very important because it really has effects also on resistant tumor cells, and indirectly on NK cells. I will go later a little bit further into this direction. In addition, Viscum album extracts can inhibit angiogenesis and may enhance the cytotoxic effects on standard anti-cancer therapy. The immunostimulatory properties are based on several mechanisms. We have DNA stabilizing effects on mistletoe extracts. We have activation of various immune cell populations, including monocytes, we have granulocytes, NK cells, dendritic cells, and T cells. At the humoral level, the induction of cytokine secretion, including interleukins such as TNF-alpha and interferon gamma, and also additional chemokines. In addition, it is also very important mistletoe extracts can promote endorphin release, which may contribute to pain release and thus to quality of life. Very important is that the effects of mistletoe extracts appear to depend on the interplay of their numerous bioactive constituents, which you see here, rather than being mediated by a single compound. Their activity results from the synergistic interactions of multiple biological active substances. So in the next few slides, I would like to discuss the scientific studies that support the hypothesis that Helixor viscum album extracts may simultaneously modulate multiple hallmarks of cancer, and by doing so through this mechanism, viscum album may contribute to transforming an immunologically cold tumor into immunologically hot tumor. Okay, let us now look at the first hallmark of cancer, which is sustained proliferative signaling. The maintenance of continuous cell proliferation is a key characteristic of tumor cells. And by activating growth-promoting autocrine or paracrine signaling pathways, cancer cells can escape normal physiological growth control. And numerous studies have shown that viscum album can induce cell cycle arrest and thereby reduce the DNA synthesis. One remarkable preclinical study in which mistletoe lectins markedly reduce the expression of amplified milk proteins, induced apoptosis, and inhibited proliferation of small cell lung cancer cells is shown in this picture. It's most important for me because I worked for years on small cell lung cancer and know how aggressive they are and how less or very few actually therapeutic approaches we have. So this apoptotic effect was also confirmed in vivo. Treatment with mistletoe lectins over a period of 4 weeks significantly reduced tumor growth in SLC xenograft model, so in human tumors in mice, both as monotherapy and in combination with cisplatin. At the same time, the treated tumors showed increased apoptosis, and as illustrated by this representative micrographs, I hope you can see it well, we can see reduced Ki67 staining. It is shown here in brown, indicating decreased proliferative activity. Ki67 is the proliferation marker, together with increased expression of cleaved caspase 3. It is shown here in red, and as you can see here, so we have apoptotic marker increase and reduction of the proliferation marker. Okay, let's go into the second hallmark of cancer. It is inducing angiogenesis. For tumor growth and metastasis, cancer cells depend on angiogenesis, right? So the formation of new blood vessels. Preclinical studies suggest that viscum album preparations can inhibit angiogenesis through several mechanisms. Not one, several mechanisms. One very important mechanism is reducing VEGF expression, which we see here, inhibiting endothelial cell proliferation, especially when you look at this part, and inducing apoptosis of endothelial cells. In this preclinical in vivo study, the effect of Viscom album on angiogenesis were investigated in adenocarcinoma xenograft, and what I want to emphasize, it was a single peritumoral injection of Helixor Viscom album, which resulted in such a tumor microvascularization reduction. So only one Helixor injection, not directly into the tumor, but peritumoral injection. And this reduced angiogenesis was accompanied by inhibition of tumor growth. Okay, let's move on to the hallmark 3, activating invasion and metastasis. Metastasis accounts for approximately 90% of cancer-related deaths. Several studies suggest that Viscom album extracts can inhibit tumor cell migration and invasiveness, so both invasion and migration. This is illustrated here in a study using a human glioblastoma cell line, like really very aggressive cell line, compared with a control group. In vitro cell migration was remarkably reduced by Viscom album. By aviscomine, it's a recombinant non-glycosylated form of methyltolectin-1, and by purified lectin-1, methyltolectin-1. The underlying mechanism appear to include modulation of adhesion molecules, for example, integrins and matrix metalloproteinases. In another study on breast cancer model, the migratory capacity of tumor cells was investigated using a transfer chamber assay, as you see here. In this study, the cells must migrate through an extracellular matrix barrier. And here again, mistletoe extracts produced a concentration-dependent reduction in cell migration. So they have, through this transfer extracellular matrix, they actively need to migrate toward the chemokine or cytokine. Okay. In a mouse model of breast cancer, mistletoe extracts significantly reduce the formation of lung metastases, as you see nicely here, both in number and in size. And in these histological sections, the metastatic tumor areas are shown in green, and as you see, there is a huge reduction in metastases, which was also associated with decreased expression of several metastasis-related genes, including MMP3 and MMP9, matrix metalloproteinases, which are involved in extracellular matrix degeneration. So this MMP3 and MMP9 are actually the very important matrix metalloproteinases. They degrade the extracellular matrix and so enable tumor cells to migrate and to metastasize, invade. Then we have UPA and UPAR, this components of the urokinase-dependent proteolytic system, and they contribute to the activation of matrix metalloproteinases. So we have here the UPA system, which is activated and also leads to the activation of MMPs. Most importantly, we have here snail and fibronectin, two important markers associated with EMT, epithelial mesenchymal transition, and which increased tumor cell migration and invasion. So especially snail is the EMT marker. It is always increased when we have EMT, which leads to the migration and invasion. And here you see very nice reduction upon Viscom album. So which means when all these markers are down-regulated, tumor cells may lose part of their mesenchymal phenotype and consequently show a reduced capacity of migration and invasion, and so metastatic spread. With EMT, we now come to the next hallmark of cancer, phenotypic plasticity. Because I already mentioned EMT on tumor cells, I would like here to go a little bit further and discuss with you phenotypic change in tumor microenvironment, because Viscom album does not only have impact on tumor cells phenotype, but also on the phenotype of tumor microenvironment. Phenotypic plasticity is actually a key feature in malignant tumors. It enables cells to switch between different cellular states. I mentioned already EMT, but in tumor microenvironment, we have also immune cells. We can switch from from anti-tumorigenic into pro-tumorigenic state of phenotype. One important example is a transition between M1 and M2-like macrophage states, often referred to as macrophage plasticity. Tumor cells release cytokines and growth factors such as interleukin-10, TGF-beta, and CSF1, colony-stimulating factor 1. And these factors can reprogram macrophages from a predominantly pro-inflammatory M1-like phenotype towards a more immune-suppressive M2-like phenotype. And these two associated macrophages can promote tumor growth, angiogenesis, tumor remodeling, metastasis, immune suppression, and resistance to therapy. So we speak here of M2 macrophages. In this study by Hong and Liu, the effect of Viscom album on macrophage plasticity was investigated very nicely and very intensively. We have breast cancer cells here cultured in three-dimensional co-culture system together with either M1 or M2-like macrophages. And they were additionally treated with Viscom album. So tumor cells with macrophages as controls and treated with Viscom album. When you compare these two pictures, M1 macrophages showed cytotoxic activity as reflected by a clear reduction in the number of viable tumor cells. And this induction of tumor cell death was further enhanced by Viscom album treatment. In contrast, when we compare M2 macrophages, they initially showed a protective effect by reducing apoptosis in tumor cells. So there is a huge difference between M1 macrophages and M2 macrophages. But interestingly, this protective effect was abolished by Helixor Viscom album treatment, and moreover, under this condition, the M2 macrophages acquired cytotoxic properties. Thus, Viscom album treatment not only enhances the cytotoxic activity of M1 macrophages, but also appeared to promote a functional reprogramming from M1 to M2, from M2 to M1 macrophages. So reactivating the tumor microenvironment for the immune stimulatory effects. let's look a little bit deeper into M1 and M2 macrophages. We have here M1 macrophages, which characteristically secrete high levels of interleukin-6. And this secretion was further increased following Viscum album treatment. Then we have M2 macrophages, by contrast, which produced relative low levels of interleukin-6. Interestingly, treatment of Viscum album increased interleukin-6 secretion, also in M2 macrophages. What does it mean? It suggests there is a shift from M2 into M1 state. And similar to this phenomenon, we also have M2 macrophages. Here, interleukin-10 secretion is important. Interleukin-10 is generally associated with immunoregulatory and immunosuppressive functions, and was only weakly secreted by M1 macrophages because they stimulate or secrete interleukin-6. But following Viscum album treatment, interleukin-10 levels were further reduced, which means these macrophages are further activated. Importantly, interleukin-10 secretion by M2 macrophages was also markedly decreased after Viscum album treatment. What does it mean? The M2 phenotype is here suppressed. So taken together, the increase in interleukin-6 together with a simultaneous reduction in interleukin-10 supports the hypothesis that Viscum album treatment can promote reprogramming from M2 macrophage into M1 pro-inflammatory anti-tumorigenic macrophages. Let us now turn to the final hallmark of cancer for today, that is evading immune destruction. Tumor cells secrete chemokines and cytokines that recruit a variety of immune suppressive cell populations, including myeloid-derived suppressor cells. We have here Tregs, regulatory T cells. We have TAMs, tumor-associated macrophages. We have T17 cells, which are T helper cells, but suppressive T helper cells. We have DCREGs, regulatory dendritic cells, tumor-associated neutrophils, and Bregs, regulatory B cells. All of them are actually immune suppressive. And together, these cells contribute to the formation of immunosuppressive tumor microenvironment. And in addition, tumors can actively alter the phenotype and function of otherwise normal immune cells, shifting them from an anti-tumor state towards an immunosuppressive state. So, two steps. First of all, when the active immune cells are recruited into the immune tumor microenvironment, they are actively suppressed by the tumor, and the tumors actively recruit inhibitory immune cells. And these immune suppressive cells then inhibit the cytotoxic function of very important cells, which are NK cells, natural killer cells, and cytotoxic CD8 T cells, both cells which have a huge impact on anti-tumor effects. And this occurs through the expression and release of variety of inhibitory factors. We have PD-L1, we have CTLA4, we have also arginase, TGF-beta, IDO, and as I mentioned, interleukin cell. And as a result, these immune cells not only lose their ability to effectively attract the tumor, but may even contribute to tumor growth and disease progression. And it is precisely within this hallmark that Viscum album appears to play a particularly interesting role, because it may help reactivate the body's own immune disease. So what we saw here, all of the cells could be again reprogrammed by Viscum album. And indeed, preclinical and clinical studies indicate that Viscum album can stimulate both the innate and the adaptive immune system. Important mechanisms: first, on the left side, the innate immune response include macrophage activation with enhanced migration and phagocytic activity. We have granulocyte activation, including improved migration, phagocytosis, and oxidative burst. We have enhanced NK cell cytotoxicity, in my opinion, the most important point. Components of mistletoe extracts include viscotoxin, includes lectins, which can increase the cytotoxic effects of natural killer cells, which are critical in eliminating tumor cells. We have then maturation of dendritic cells, thereby improving their capacity for antigen presentation. And here we have the connection from innate to adaptive immune response. Important mechanisms of the adaptive immune response include proliferation and activation of T cells, particularly CD4 helper T cells, but we have also CD8 cytotoxic T cells, both of which are important in anti-tumor immunity. There is also evidence that Viscum album also regulates Treg cells, regulatory T cells. We have proliferation of mistletoe-specific B cells and the production of antibodies against mistletoe lectins. We have increased secretion of cytokines associated with adaptive immunity, such as interferon gamma, and in parallel, increased the release of cytokines associated with innate immune activation. We have here interleukin 1, interleukin 6, 10 of alpha, which all may contribute to the establishment of more immunologically active tumor microenvironment. Okay, with this, let's have a closer look at NK cell activation. May I just ask, Dr. Joel, you still hear me very well, right? Should I continue this way?
Excellent. Everything is perfect. Excellent.
Perfect.
And translation is working perfectly.
Wonderful. Thank you very much. Thanks. Okay, so then I would continue with NK cell activation. This is one of my favorite studies shown on Helixor A effect on natural killer cells. Here, primary NK cells were isolated from the peritoneal fluid of patients with endometriosis. As a control, NK cells or primary NK cells were obtained from the peritoneal fluid of patients with benign ovarian cysts. So we have control NK cells from ovarian cysts, peritoneum, and the study was actually on endometriosis. A total of 100 samples were analyzed with 15 samples in each group. NK cell activity, in other words, cytotoxicity, was assessed using Ki562 cells, a human CML cell line. It was used as a target cell. As a reference control shown here on the left side, we have NK92 cells. This is a natural killer cell line. This showed the cytotoxicity of approximately 80%. A similar level of cytotoxicity was observed with NK cells isolated from peritoneal fluid of the control group. But in contrast, NK cell activity was lower in the endometriosis group, and cytotoxicity decreased with increased disease stage. So in patients with stage 1 and 2 endometriosis, NK cell cytotoxicity reached only about 70%. In stage 3 and 4 disease, it decreased significantly further to approximately 64%. So these findings indicate that NK cell cytotoxic function is impaired in patients with endometriosis, particularly in more advanced stages of the disease, which means it is very comparable to the cancer patients. The effect of Helixor A on NK cell cytotoxicity was investigated. For this purpose, the cells were incubated with 200 nanogram per milliliter Helixor A for 24 hours and subsequently assessed for cytotoxic activity. And all 3 groups, NK cell activity was significantly increased following treatment with Helixor A. And interestingly, the strongest effect was observed in the group with a stage 3 and 4 endometriosis, where NK cell function has been most impaired at baseline. So the stronger the impairment, the more effective viscum album seems to be. So this naturally raised an important question. Does viscum album simply activate NK cells, or could it also exert inhibitory or even pro-apoptotic effects on the cells? So to exclude this possibility, an apoptosis assay was performed. We have here NK cells where the cells were incubated with viscum album for 24 hours and subsequently analyzed by FACS, by flow cytometry. And the dot plots allow us to clearly distinguish the different cell populations. We have in the lower left quadrant, we see the viable cells. These cells are double negative for both for annexin 5 and for propediumidate. So both actually indicate apoptosis, both are negative. So we have here our viable cells. In the lower right quadrant, we see here the cells are in early apoptosis. These cells are annexin 5 positive, but still propedium iodide negative, because propedium iodide goes into the DNA and binds DNA. So the cells are not that apoptotic so that propedium iodide could bind here. Then we have in the upper right quadrant the double positive cells. This represents the cells which are late apoptotic or even necrotic. So both stains are positive. And in the upper left quadrant, we see predominantly necrotic cells. So the membrane, the annexin 5 is already gone, and we have propidium iodide positive cells. So as you can see, treatment with Viscum album did not increase the proportion of apoptotic cells, which is very important. This means the NK cells are very active, very cytotoxic, but remain their viability during Viscum album treatment, indicating that the observed increase in cytotoxic activity was not accompanied by increased NK cell apoptosis. The mean overall proportion of apoptotic NK cells also remained unchanged following the treatment with Helixor A. Now, NK cell activity was assessed functionally by measuring cytotoxicity. But in addition, in most studies, NK cell activation is also analyzed at the phenotypic level. A commonly used marker for this is CD107A, and CD107A is associated with cytotoxic granules and becomes transiently exposed on cell surface during degranulation, and therefore it serves as a marker in NK cell activation and cytotoxic activity. Following incubation with Helixor A, surface expression of CD107 increased approximately twofold in all three groups. And this further demonstrates that incubation with Viscum album leads to activation of NK cells. Based on these findings, the authors proposed that combining Helixor A with standard therapy may represent a potentially effective therapeutic approach for endometriosis, but also for cancer patients, because it's a similar mechanism. Okay, now I would like to show you very shortly the antiviral effect of Helixor Viscum album on the case series. It's not a clinical study, but a case series on HPV positive patients. And in this case series, 7 outpatients with persistent HPV virus were treated with subcutaneous Helixor injections and additionally topical Helixor treatment. So we have both. We have subcutaneous Helixor injection, Helixor M, and topical Helixor treatment. After the therapy, 6 out of 7 patients had negative hybrid capture test for HPV. Thus, Helixor M shows also antiviral effects. So let us now turn to the clinical evidence, specifically to the safety and tolerability of Helixor therapy, especially in combination with immune checkpoint inhibitors. In the next steps, I would like to highlight 3 studies which perfectly fit to show the whole treatment of Viscum album with immune checkpoint inhibitors regarding or viewing at it from different points of view. So we have here, the first study. It is a real-world data study investigating Viscum album in combination with immune checkpoint inhibitors and other standard anti-cancer therapies. So we speak actually only with immune checkpoint inhibitors, but actually patients also had all the other standard anti-cancer therapies. The study analyzed oncological patients who received either immune checkpoint inhibitors alone or in combination with immune checkpoint inhibitors and Helixor viscum album extracts. All patients also received conventional oncological treatment, including chemotherapy. We have, in some cases, combination with radiotherapy, hormone therapy, or surgery. Among 405 cancer patients, 344 received immune checkpoint inhibitors alone and from the control group, while 61 received the combination of immune checkpoint inhibitors and Helixor viscum album. Two questions were addressed. First, the tolerability of the combination therapy, and second, the effectiveness of the combination therapy, which was evaluated analyzing overall survival in patients with non-small cell lung cancer. So the first step, the tolerability, here were included several tumor entities, and then to be more specific, the authors went with a focus on NSCLC. In addition, overall survival was analyzed separately. Was analyzed separately in the female subgroup with a particularly interesting result that I will come back to shortly. So, as I mentioned,
fui a consultor para que me lo revisaran y ya me creció bastante.
Can you hear me? Yes, right?
Yes, yes, yes. It was a problem with the one that activated the microphone. Don't worry.
Okay, wonderful.
Keep ahead.
So I will continue. Thanks. Regarding the tolerability analysis, this study has three major strengths. First, it included a broad spectrum of different tumor entities. As you can see, the most frequently represented cancer types was lung cancer, particularly non-small cell lung cancer, accounting for almost 79% of patients. This was followed by breast cancer at 7.2%, malignant melanoma, and renal cell carcinoma. In addition, several other tumor entities were included, such as Hodgkin lymphoma, colorectal cancer, hepatocellular carcinoma. So very important, a broad range of tumor entities. Second point, we had a broad spectrum of immune checkpoint inhibitors. Three major classes of immune checkpoint inhibitors were represented. We have PD-1 inhibitors accounting for 71.1% of treatments, PD-L1 inhibitors accounting for 25.4% of treatments, and also CTLA-4 inhibitors. Third important point is only Helixor viscum album preparation were used. So this helped to reduce product-related heterogeneity and allowed the effect of viscum album to be assessed as clearly, as consistently as possible. So the authors only concentrated on patients who had nothing else as Helixor viscum album treatment. Because the study included a wide range of tumor entities as well as a broad spectrum of immune checkpoint inhibitors, it allowed for a differentiated assessment of treatment tolerability. In the overall cohort, 25 of the 405 patients, corresponding to 6.2%, discontinued treatment due to immune-related adverse events. In the control group, 22 patients, or 6.4, shown here in gray, discontinued treatment because of adverse events. So we have here 6.4 in control group and 4.9, only 3 patients in the Viscum album group. The difference between the two groups was not statistically significant because we concentrated on a small number of patients, but still what I would like here to emphasize, it is not the number or only the number of adverse events, because adverse events were really high, the difference. What we the authors analyzed was the adverse events which really led to the discontinuation. That it was so strong that it led to the discontinuation of the therapy. So it is important to emphasize that this study did not assess the overall incidences of adverse events, as I mentioned, but only the discontinuation. And based on these findings, the authors proposed that Helixor Viscum album therapy may help reduce treatment-related side effects and thereby potentially improve adherence to standard oncological therapy. Regarding the safety, I would also like to mention an additional finding, especially regarding the liver parameters from a recent study that had not yet been published. In this multi-center analysis, 68 patients from 4 different centers in Switzerland were evaluated with regard to their liver function parameters, and only 9 patients showed mildly elevated liver enzyme levels, and importantly, these elevations were not considered to be related to Viscum album therapy. All the patients, why did we actually analyze the liver enzymes or liver parameters, is that all these patients, all the 68 patients were treated with Helixor Viscum album in very high dosage. So when we speak about high dosage, it's between 2 gram and 4 gram. And still in this high dosage, no toxicity in liver parameters. Okay, so having looked at safety, let us now turn to efficacy. In the same study, in the next step, the efficacy of immune checkpoint inhibitor therapy with and without Viscum album was analyzed. To obtain more robust and clinically meaningful results, a subgroup analysis focused on patients with non-small cell lung cancer was performed. So now we narrow focus only on NSCLC. The survival analysis showed a significant survival benefit for the combination group, shown here in red, compared to the control group, shown in green. The 3-year survival rate was 34.3% in the combination group, which was approximately twice as high as a control group, where it was only 17.2%. The difference was statistically significant. Then the authors looked for 5-year survival rate, which was 22% in the combination group, approximately 2.5 times higher than the 8.8% observed in the control group. Interestingly, the survival benefit was even more pronounced in the female NSCLC group, which included 137 patients. The 3-year survival rate was 66.8% in combination group compared with 26.9% in the control group. And at 5-year, the survival rate was 15% in the combination group, approximately 4 times higher than the 12% observed in the control group. And also this difference was statistically significant. So in the female NSCLC combination group, median overall survival was not reached during the observation period, likely because of the small sample size, but I know that the authors still are observing the median overall survival of the patients. And in this subgroup, in the female subgroup, the hazard ratio was also analyzed using a multivariable Cox proportional hazard model with the immune checkpoint inhibitor only group serving as a reference. That's why it is not shown. So it is a reference. A hazard ratio of 1 indicates no difference in the instantaneous risk of an event between 2 groups. A hazard ratio below 1 indicates a lower event rate in the treatment group, whereas a hazard ratio above 1 indicates a higher event rate. And in this analysis, the combination therapy was associated with a statistically significance of 91.2% reduction in the risk of death compared to the control group. So group immune checkpoint inhibitors with Viscum album compared to only immune checkpoint inhibitors. Importantly, this effect remained significant after adjustment for age, use of therapy in the first-line setting, and tumor stage. So, in summary, all these results suggest that in patients with NSCLC, the combination of immune checkpoint inhibitors with Helixor Viscum album is associated with safety and improved survival, especially for female patients. Next, I would like to present another study addressing a similar question. This real-world data study, also conducted by Dr. Schad and his research group, was published 1 year earlier, so in 2024. It included patients with advanced and metastatic non-small cell lung cancer. So the previous study was conducted or included all NSCLC patients, and this one specifically advanced and metastatic non-small cell lung cancer. The study analyzed overall survival in patients treated with and without Viscum album therapy. So the study included patients with NSCLC who received either both or immune checkpoint inhibitors. We had here Viscum album extracts from all 3 manufacturers, Helixor, Iscado, and Abnoba. All patients also received conventional oncological treatments. Among the 415 patients with NSCLC, 222 received immune checkpoint inhibitors alone and formed the control group as a previous study, while 193 received the combination of immune checkpoint inhibitors and viscom album therapy. The first focused analysis was on overall survival in patients with advanced and metastatic NSCLC, and then the authors went a little bit further subsequently and analyzed a subgroup which had a patient receiving first-line therapy with PD-L1 positive tumors. Similarly, also here, the addition of mistletoe therapy was associated with a significant improvement in overall survival. So we have here median overall survival in the control group, shown here in black. It was 6.8 months. In the combination group, shown in green, the median overall survival increased by 17.8 months, an improvement of approximately 7 months. In addition, a hazard of death was reduced by approximately 40% in the combination group. This survival benefit was even more pronounced in the subgroup, which I mentioned before. In this subgroup, median overall survival was significantly prolonged by approximately 21 months. So not 7 months, but 21 months now. Median overall survival in the control group was 5.4 months, whereas the combination of immune checkpoint inhibitor therapy with mistletoe therapy increased median overall survival to 26.4 months. The 1-year survival rate was 66.3 in the control group compared to 34.4 in the control group. So combination group 66.3, control group 34.4%. At 5 years, the survival rate was 16.5% in the combination group, nearly 3 times higher than the 5.7% observed in the control group. In addition, again, we had here the hazard of death was reduced by 56% in the combination group compared to the control group, which was again immune checkpoint inhibitor group. To sum up, again, the addition of mistletoe therapy, and in this time we had all 3 products just to emphasize our Helixor results. Also, this study showed a significant improvement in overall survival. Now, I would like to share with you some unpublished data